Today on In Your Face Anatomy, we are going to learn some muscles. Here we go. Muscles. So, a quick review. With muscles, we learned this when we did tissues back in chapter 4. So remember, we have 3 different types of muscle tissue. I have skeletal muscle. This is what's attached to bones. It is voluntary, which is crazy important. It's our voluntary muscle, multinucleated, and it is striated. So remember, striations are the pattern and the repeating pattern that we see when it comes to tissue. So skeletal muscle has that repeating pattern, so it is striated. Then I've got smooth muscle, which is kind of the opposite of skeletal muscle. Smooth muscle we find in the walls of our viscera, our guts, blood vessels, skin. It is involuntary, meaning we have no conscious control over this muscle. Our brain is kind of controlling it in the background with us having to give it specific thought. It has a single central nucleus, and it is non-striated. So its filaments get laid down randomly, remember, so we don't get that same repeating pattern that we see with skeletal muscle. And then lastly, we have cardiac muscle. As the name will suggest, we find this in the wall of the heart. It is involuntary, like my smooth muscle, but it is striated, like my skeletal muscle. And with our cardiac muscle, remember, we also have intercalated discs that help connect the muscle tissue together, makes it easy to spot and once you know what you're looking for with those intercalated discs. So 3 basic different types of muscles. We're going to spend the majority of our time talking about skeletal muscle. We will touch a little bit on smooth and a little bit on cardiac, but most of this is going to be all about skeletal muscle. All right, so fascia is a sheet of fibrous connective tissue that encloses the muscle. And it can continue down, and it may form part of a tendon or an aponeurosis. So that's just kind of like the wrapping around the muscle. A tendon is a cord-like or band-like, rope-like mass of tissue that connects the muscle to a bone. So tendons connect the muscle to a bone. It's kind of the tapering end of your muscle. The attachment point is the tendon. An aponeurosis is just a different type of way to attach muscle. So an aponeurosis is a sheet of connective tissue that attaches bones, or attaches muscle to bone or to fascia. You can see in our picture here at the top of the head where the muscle kind of turns white as the skull starts to present itself. That white that we see is the aponeurosis, the attachment point of those muscles to the skull. So it's a sheet-like attachment instead of a rope-like attachment. Rope-like attachment is a tendon, sheet-like attachment is an aponeurosis. Now, when we look at the muscle, we've got a couple of different layers here. I've got the epimysium. This is what we find directly under the fascia, and it completely surrounds the muscle. So if we jump over to our picture, you can see that kind of that white outline that goes all the way around, just the outer edge all the way around. That is my epimysium. Then we have our perimysium, which is going to separate muscle into smaller sections called fascicles. Back over at the picture, you can see where the perimysium cuts in and divides that muscle up into smaller sections. Each one of those sections we call a fascicle. And if we pull that fascicle out, like we did in the bottom of our picture here, we can see the endomysium, which separates that fascicle into smaller sections, specifically into the individual muscle fibers itself. So the endomysium separates the fascicles into smaller sections. Perimysium, we already did that. Fascicle, group of muscle fibers. Muscle fibers are the muscle cells. So a muscle fiber is a muscle cell. One muscle cell is a muscle fiber. We have myofibril muscle cell organelles. They form bundles of myofilaments. And then the myofilaments are the muscle proteins themselves. So we have 2. We've got actin, which are the thinner ones, the thin filaments. And then the myosin are the thick filaments. So if we cut over to our picture here, we can see the filaments at the very end. The little blue one, that's the thinner one of the 2, so that's going to be my actin. Where the thicker one, the purple one, is going to be my myosin. And we can see how we bundle those together to make a myofibril. A myofibril is one part of the separation where we pulled in our endomysium and then those muscle fibers, we can see how they come back together to form the fascicle, which goes back up into all of the muscles. And we got a little diagram over on the right hand side that helps break it down for you so you can see what exactly is going on. So some terminology just to remind you of: sarco is muscle in Greek, myo is muscle in Latin. So sarco or myo just mean muscle. And again, we said a skeletal muscle fiber is a muscle cell, but we've got a few tweaks with this one. We've got a sarcolemma, which is what we call the muscle cell membrane. Sarcoplasm is what we call the cytoplasm in a muscle cell. We've got a sarcoplasmic reticulum instead of the endoplasmic reticulum. But we have a new structure called the transverse tubule. You'll also see this as the T-tubule quite a bit. It lies in between two cisternae of the sarcoplasmic reticulum and extends from the sarcolemma to deep within the muscle. Together, these three come together to form what we call a triad. But we can see in the middle, that transverse T-tubule of the triad in our picture, the lighter part in the middle, it goes down deep into that muscle layer, which is going to be important coming up. We've got our myofibril proteins, contractile fibers in the muscle cell walls. Actin, we said, was a thin one. Myosin is a thick one. I always just rhyme thin, actin, so then I know that's the thin one, so myosin has to be the thick one. And it's the organization of these filaments that give us the striations. And striations form a repeating pattern we call a sarcomere. So a sarcomere is defined as the repeating unit between two Z-lines. It is the smallest unit of contraction. So that's something we might want to know. The sarcomere is the smallest unit of contraction. So if we drop down to our picture, what we're going to see is we have this light grayish blue area, then we got a dark purple, we got a pink, then the dark purple, the gray, dark purple. But we can see how this pattern repeats. Every time it repeats, each one of those we call a sarcomere. And it's this repeating pattern that gives us the striations. Now, first thing, the Z-line separates sarcomeres. Or I shouldn't even say separates, it combines sarcomeres. It sticks one sarcomere to the end of the next sarcomere, and then the next sarcomere, and then the next sarcomere. And separating the sarcomeres is the Z-line. Gets its name from that zigzag pattern, so it makes it pretty easy to spot. The I band is the light band, and when I say light band, I mean light in color. So that is going to be the grayish blue area. And it's in this area that we only find the thin actin filaments. They're only the thin filaments, so that's why it's the lighter in color, because we just have these little thin filaments laying down. Only the actin is in there. The H zone, the pink in the middle of our sarcomere, that is where we find only our thick filaments. So only the myosin is in that area, in the H zone. And then the A band is the dark band, and that's where we find our thick filaments and our thin filaments. So where it's purple, that darkest purple area, that's where we have thin and thick filaments overlapping. We have both actin and myosin in that middle section where it's the darkest. So we got them both, so it gives it that darker coloration. Now I remember this because the word light has an I in it, so I band, I can do light band. The A band, the dark has an A in it, so A for dark, I for light, and that's how I can keep those straight in my head. An M line holds our thick filaments in place. We can see in this other picture now, coming down the middle there, our M line just helps hold everything where we want it to be. And like I said, a sarcomere extends from one Z-line to the next. So, myosin are thick filaments, and what makes them special, they can form cross bridges that bind to actin filaments. So that's going to be important in contraction, which we have coming up. So myosin can form cross bridges. That's what we want to know. It can bind to the actin. The actin, the thin filaments, we've got two proteins that we want to know for actin when it comes to skeletal muscle, because that's what we're talking about. I've got troponin and tropomyosin. Troponin and tropomyosin. Now these proteins lock the actin filaments and prevent myosin from being able to bind to it. So myosin can't form its cross bridges with troponin and tropomyosin in its way. That way we're not actively or accidentally activating our muscles and contracting our muscles all of the time, because we have these two proteins guarding the binding sites on actin, so myosin cannot form its cross bridges until we want it to. So the neuromuscular junction, this is our segue into our last chapter on the nervous system. So you'll see this as the neuromuscular junction or the myoneural junction, just depending on which system you're talking about. If you're talking about the nervous system, then we typically call it the neuromuscular junction. If we're talking about the muscular system, we call it the myoneural junction. But either way, they're really interchangeable. But this is where the nervous system, the axon from a neuron, and your muscle fiber meet. So the neuron is the functional cell of the nervous system. The axon is a piece of the neuron that conducts the impulse. So a neuron has a long tail on it, and that tail is the axon. So it gathers up the information and it sends that information down the axon to the next neuron, or in this case, the muscle cell, wherever our end goal is. That's how the axon works. Now, the muscle fiber membrane itself, that sarcomere, forms a divot we call the motor end plate. And we can see that in the picture. See how we've got that neuron, the end of our neuron, our axon specifically, popping down into the muscle, but there's a little divot in the muscle for that neuron to fit into to make it easy to transfer the signals from our nervous system to the muscular system. The synapse is the functional connection between a motor neuron and a muscle cell. So notice we said functional connection here at the synapse, because if you look, you can see there's a little gap that goes all the way around in our picture here, all the way around that neuron. So the neuron and our muscle cell don't actually touch. We have that gap separating the two. Now we communicate between the two, between our nervous system and our muscular system, via neurotransmitters. The synaptic cleft is the small gap that separates the neuron from our muscle cell. So the gap in between the two, we call that a synaptic cleft. Motor unit, single motor neuron, and the muscle fibers it controls. We get anywhere from 4 to thousands of muscle fibers. We get neurotransmitters, chemicals that an axon end secretes that stimulate a muscle fiber to contract. So lots of neurotransmitters. We're going to talk about a bunch of them in the next chapter. But one that we want to know for now for sure is acetylcholine. Acetylcholine, also known as ACH, is the neurotransmitter that controls skeletal muscle contraction. Neurotransmitter that controls skeletal muscle contraction, ACH. And we store this in our synaptic vesicles. So now that we have all the pieces in place, let's talk about how a muscle actually contracts. So we want to know this, and we want to know this in order for skeletal muscle. So a nerve impulse runs down our axon from our neuron, hitting the synaptic vesicles at the end of the axon. That is the signal for the neuron to release ACH, acetylcholine, into that gap, the synaptic cleft, in between my muscle and my nerve. So the nervous system sends an impulse down the neuron all the way to the end of the axon. That's where we've got our little neurotransmitters stored, and that signal releases those neurotransmitters into the gap, the synaptic cleft. ACH is then going to move across that gap and bind to receptors in the muscle fiber membrane. This allows sodium to rush into the muscle, and that depolarizes the membrane. Because sodium has a charge, it's an ion. We can see that Na+. So moving that in is going to mess with the charge on the muscle fiber membrane, cause it to depolarize as more positive charges rush in. This action potential produces the membrane to depolarize, and it travels down the T-tubule deep into the muscle, which causes a lot of calcium ions to be released into that sarcoplasmic reticulum. So the sodium rushes in, and that causes calcium to get released. Now these calcium ions bind to troponin, causing tropomyosin to shift positions and expose their binding sites. So when calcium rushes in, what it does is it peels back that troponin and tropomyosin that's blocking myosin from binding to it. But once we peel it back and those binding sites are revealed, Now we can use ATP to get myosin to form those cross bridges, to bind to actin, and then the myosin bends, which slides the actin over the myosin for contraction. So that's how the muscle actually contracts. We need sodium ions to come in to release the calcium. We need calcium to come in to remove those proteins, troponin and tropomyosin, that are protecting the binding sites. Once those binding sites are exposed, myosin forms its cross bridges, binds to actin, and then they slide past each other. So, muscle contraction, when the sarcomere shortens, our thickened filaments slide past one another. We can kind of see that in our pictures over here. As they slide, our H zones and our I bands are going to narrow. The Z lines are going to move closer together. This causes the sarcomere to shorten, and we call this the sliding filament theory. So when a muscle contraction occurs, nothing really shortens. We say that kind of as default because it's easy to visualize, but the muscle doesn't actually shorten. What happens are the filaments slide past each other, causing the muscle to appear like it's shortening, and we can see that in our picture between a relaxed muscle and a fully contracted muscle. All right, so now that we've contracted the muscle, it's time to get that muscle to relax. And we do that with an enzyme called acetylcholinesterase. So remember, we know it's an enzyme because it ends in ase. Acetylcholinesterase. So this is the enzyme that's going to break down acetylcholine. So it rapidly decomposes any ACH remaining in the synapse. This is going to stop that muscle impulse. It's the stimulus to the sarcolemma. Muscle fiber membrane ceases now. Calcium moves back into that sarcoplasmic reticulum, which now prevents myosin and actin from binding together, and in result, the muscle fiber relaxes. So to make this happen, we've got two different types of energy sources that we can use for contraction. Cellular respiration, 1, and 2, creatine phosphate. So cellular respiration, creatine phosphate are our main energy sources. So cellular respiration is the main energy source. That's the one that we use primarily for pretty much everything that we do. So this is the citric acid cycle, or the Krebs cycle. You've probably seen that before in a biology class or in high school, where we take one sugar and we turn it into 32 to 34 molecules of ATP, which we could then use as energy. So what we're doing with cellular respiration, we're converting glucose to ATP, turning sugar into energy. Muscle stores glucose in the form of glycogen. So that's one way we control our sugar in our bodies, is we just turn in sugar into glycogen, and then we store that in our actual skeletal muscle. So it's right there ready for us as an energy source for when we go to move. We simply turn glycogen back into glucose and then we break it down into the ATP that we need. So that's cellular respiration. The main energy source you want to know, Krebs cycle, converting glucose to ATP and storing glucose in the form of glycogen. All of that is super important stuff when it comes to energy sources for actually contracting the muscles. The other energy source, creatine phosphate, is kind of a muscle or an energy storage process, I want to say. It stores energy that quickly converts ADP into ATP, but it's not sufficient to support maximal muscle activity for more than 10 seconds. So I always think of this as kind of like my oh shit energy. So if you're outside and you walk up on a snake or a bear, we'll say, you walk up on a bear and it scares you, you get that rush of energy for your muscles to work for about 10 seconds, but it's that burst of speed that you need out of the gate to get you out of the situation, away from the bear, and then your body's going to kick over into cellular respiration, but you have that kind of 10 seconds reserve energy ready to go at all times in your body. It's just an evolutionary adaptation from back when we were hunter-gatherers. Muscle fatigue is defined as the inability to contract a muscle, commonly caused from decreased blood flow, ion imbalances across the sarcolemma, accumulation of lactic acid, cramp, sustained involuntary muscle contraction. A twitch is a brief muscle contraction followed by relaxation. So, a couple of good definitions in here. Let's absolutely know the difference between a cramp and a twitch and what can cause your body's inability to actually contract a muscle. So muscle responses. So we got a couple here. So threshold stimulus is the minimum strength required to cause contraction. Let me read through these real quick and I'll come back. Latent period, time between an application of a stimulus and the beginning of a response in a muscle fiber. Contraction, we've already talked about. Relaxation, we've already talked about. We got a refractory period, the period following stimulus during which a neuron or muscle fiber will not respond to a stimulus. And then we want to know it's an all or nothing response when it comes to muscles and nerves for that matter. So what this means is there is a minimum strength that is required to signal a muscle to contract. We have to get to that point. And I want to say it's negative 70 millivolts is what I want to say. Negative 70 millivolts. That's going to be in the next chapter, I think. I don't think it's in this one. Yeah. So we have to hit that. That's our threshold. We have to put that much effort in to get a muscle to contract. If we do negative 69 millivolts, no, it's not going to contract. If we do negative 72, it absolutely will because we're above that threshold. We have to at least get to negative 70 millivolts. We have to meet that minimum strength requirement. So that's why we call that the threshold stimulus. So the threshold is just the point we have to get to get that muscle contraction to even start. Now, once we reach that threshold stimulus, we then enter the latent period, and that is the briefest amount of time between reaching that threshold stimulus and the start of the muscle contraction. There's that brief pause. We refer to that as our latent period. Then we roll into the muscle contraction, and we go through that whole process of sending the impulse down the neuron and getting that muscle to contract. Then the muscle relaxes. We send in acetylcholinesterase to break down any ACH that's still there so we can stop the contraction, and the muscle relaxes. After that, we enter the refractory period. So the refractory period I want you to think of as the reset period, or the reset time it takes to move all of the pieces back in place so that muscle contraction can occur again. So it's resetting the period following stimulus during which a neuron or muscle fiber will not respond to a stimulus because we haven't moved everything back into place. That's our refractory period. So once we get that, then the whole process can start over and we can go back into the threshold stimulus if we need that muscle to contract again. Now, the all or nothing is you can't partially contract a muscle. Either we hit that threshold stimulus and we get that muscle to contract, or we don't. So it's all or nothing. We have to get that process, we have to meet that threshold to even get it going. So I hope that explains that, and I would absolutely know the definitions for these. These are just super easy test questions to write, so I would 100% know those. Now, when the muscle contracts, we have two different types of muscle contractions. I've got isotonic muscle contractions, and I got isometric muscle contractions. So I'm going to start with isometric because I think that's just the easier one. What we want to know is iso means same, and metric, we're talking about length. So this is the same length. So the muscle contracts, but it does not change lengths. And we see that in certain exercises like the plank that like this guy is doing in our picture here. He's got his ab muscles. Some of his glutes are probably, and his hamstrings are probably contracted as well, but he's just holding that position so the muscle's contracted, but we're not making the muscle shorter or longer. We're just holding it in that position. That is an isometric contraction, same length contraction. Now, an isotonic muscle contraction, that's where the muscle contracts and it does change length. So we can have either a shortening contraction or we can have a lengthening contraction. So I think the easiest way to visualize this is like trying to do a pull-up on one of the pull-up bars. The shortening contraction is called a concentric isotonic contraction. And so that's where we'd be pulling our chin up over the bar, and we've shortened all of those muscles in our biceps specifically for that contraction to occur. Then when we release and we drop back down, if we're doing it slowly, that's our eccentric isotonic contraction. That's the lengthening contraction. So it's still contracted, but the muscle's getting longer. All right, smooth muscle real quick. So compared to skeletal muscle, smooth muscle is shorter. We've got that centrally located nuclei. Again, No striations because our actin and myosin filaments get put down randomly. It doesn't have T-tubules, and its sarcoplasmic reticula is not well developed. Now we have two types of smooth muscle I need you to be able to distinguish. One, visceral smooth muscle, and two is my multi-unit smooth muscle. Visceral smooth muscle and multi-unit smooth muscle, and we're going to look at each of those separately. So first up is my visceral smooth muscle. So remember, viscera means your guts. So that's where we find this, in the walls of most of our hollow organs. We get sheets of muscle fibers, and we have some gap junctions in here as well. But the big thing I need you to know about visceral smooth muscle is it exhibits both rhythmicity and peristalsis. So rhythmicity, predictable muscle contractions in a repeating pattern. Like your heart has that bump, 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 that heartbeat. That is a rhythmic contraction because it's a repeating, predictable pattern that we can associate with our heart. We also have that in some of our smooth muscle, our visceral smooth muscle specifically when it comes to our organs. So we got rhythmicity, and then peristalsis is a wave-like muscle contraction that we use to propel substances forward. We see this primarily in things like our intestines, where we can contract it and then relax it, creating this wave-like effect that moves through our entire intestines. And anything on the front side of that wave is going to get propelled forward or pushed forward a little bit, but that's how we're able to move our food through our intestines for absorptive capabilities. So visceral smooth muscle, rhythmicity, peristalsis, walls of most of our hollow organs. Those are the big things that we want to know. Now, multi-unit smooth muscle is where our fibers function separately. We're not working as a group. We're functioning separately now, less organized, separate units. We see this in things like the iris of the eye and in the walls of our blood vessels. So when you cut yourself, if you cut your finger, the first thing your body does is vasoconstrict that blood vessel, just meaning it makes that blood vessel smaller. And in doing so, that means the amount of blood that we're losing becomes less because the hole leading out of the blood vessel is now smaller, so we're bleeding out less. What we don't want to do is cut ourselves and have every blood vessel in our body get smaller in response to this cut on our finger. So instead, we use multi-unit smooth muscle where we can kind of individualize the muscle and contract what we need to contract. So multi-unit smooth muscle, fibers function separately. I would know iris of the eye, walls of blood vessels functioning as separate units, something along those lines. Now, smooth muscle resembles skeletal muscle contraction in that we still have actin and myosin. We're still using calcium and ATP, and both are triggered by the membrane impulses. So everything we learned with skeletal muscle contraction, I shouldn't say everything, but some of the things we learned with skeletal muscle contraction also apply to smooth muscle contraction with a few exceptions. One, in smooth muscle, we don't have troponin. Instead, we use a different protein called calmodulin and tropomyosin in smooth muscle. Skeletal muscle is troponin and tropomyosin. We use two different neurotransmitters for smooth muscle. We're still using ACH like we did with skeletal muscle, but now we're also adding in norepinephrine as well. Hormones can have an effect on smooth muscle. It's slower to contract, but it's also slower to relax. It's more resistant to fatigue, and it can change length without changing tautness. Cardiac muscle, of course, in the heart, its fibers are joined together by intercalated discs, which we can see in our picture here, that darker line cutting through the tissue. Those are our intercalated discs connecting different sections of cardiac muscle together. Its fibers contract as a unit, self-exciting and rhythmic. We already talked about the rhythmicity. And it has a longer refractory period than skeletal muscle does. So it takes a little bit longer to reset after each contraction with cardiac muscle than skeletal muscle. All right, bones and muscles together interact as levers or levers. These are simple mechanical devices composed of 4 parts, and we're going to compare each of these 4 parts to our body. So the basic 4 components, I've got a rigid bar, which would be equivalent to the bones inside of our body. A fulcrum, the point on which we can get the bar to move. So in our body, that's going to be the joint. the object, whatever we're trying to move with the fulcrum, moved against resistance. So that's the weight. That's whatever we're trying to pick up, whatever we're trying to do, whatever we're trying to push or pull. And then the force supplies energy for the actual movement itself, and that is our muscles. So for this slide, I would know the 4 basic components of a lever, meaning the rigid bar, the fulcrum, the object, and the force. Then I would know that the rigid bar is the bones, the fulcrum is the joint, the object is the weight, and the force is the muscles. All right, origin and insertion. So we learned this in the last chapter. Origin is the immovable end or the fixed end of the muscle, where the insertion is the movable end. Now typically our muscles work in groups. So I've got my prime mover. This is also known as the agonist. This is what is primarily responsible for whatever movement is we are trying to do. It's the one that's doing the lion's share of the work. So in our picture here, we've got this guy. Let's pretend he's got a weight in his hand here, so he's struggling to pick it up. So the prime mover in this case is going to be the biceps muscle, biceps brachii specifically, doing most of the work. But he's also going to have some synergists muscles that help him pick up this weight. They assist the prime movers, what our synergists do. So I have muscles like brachialis that are under this muscle, or coracobrachialis running down the side. Any of the surrounding muscles, they're going to help. But at the same time, I also have an antagonist muscle working. So this is going to resist whatever my prime mover's action is. It causes movement in the opposite direction as the prime mover. And I know you're thinking, why would we have an antagonist resisting what we're trying to do? If we're trying to pick something up, why do we have a muscle working against us? Well, that is so the muscle doesn't contract too far and completely eat up, destroy your joints. So if this biceps flexes all the way back, that's going to dislocate the elbow. So in this case, it would be our triceps brachii running along the back of the arm, which we can't see in the picture, but it runs on the opposite side of the arm, and it's going to wrap around and it's going to stop that the biceps brachii from flexing too far up as you're trying to pick something up. So it's there just to help protect your joints. So know that the prime mover is known as the agonist, and it's primarily responsible. Synergists help the prime mover, and then the antagonist resists the prime mover. Okay, yeah, another picture here. Oh, I should have used this picture where it shows some of the other muscles. All right, but it's the same information. I'm not going through it all again. You can look at this picture on your own. All right, and that is it for muscles. We'll go through muscles to know at a later date and in class with the models. Good luck.